main goal of our project is to test a new way of treating disease called interception. Interception means we would try to go back in time and catch a disease at the very beginning before it starts. And this means we need to identify the very first disease cells before we get symptoms and find ways of removing them from the body, making us stay healthy. Here in our clinic and many clinics all over the world, we have elderly patients coming to us, and they're actually weak, they get a lot of infections, and eventually many of them, actually 1 in 3, will develop leukemia, which is a deadly disease. And there's very little what we can do about this now. So what we want to do is to travel back in time to identify these diseases and these patients before they reach the severe symptoms and treat them there. We want to reach our goals by training the next generation of European researchers, to enable them to actually put together latest technologies and the knowledge about these diseases, and to work together between industry, academia and clinic to actually make this happen.
We will try to analyze which are the cells in the myelodysplastic syndromes and in sporadic acute leukemia that are important to initiate the disease. We will try to check which are the specific populations of cells that accumulate the driver mutations. We are going to use a very ambitious experimental model ranging from primary samples from patients to cell line models, together with a state-of-the-art technology to analyze the molecular biology beyond this, such as single-cell analysis.
In this work package, we will use human cell lines, primary cells from human patients, as well as mouse models to investigate the pathogenesis of MDS with a specific focus on epigenetic processes. The projects in this work package will use genetic tools, but also pharmacological interference with epigenetic pathways to interrogate deregulated epigenetics in MDS, to reach a better understanding of the pathology of MDS and to find eventual ways to target this disease.
So in this project, we are going to be generating new drugs to fight against MDS and ALL, and we will be also generating a lot of molecular data, right? So in work package 3, what we will do is develop tools that support this development so that we can better analyze this data. So to reach these goals, we are going to be doing basically 2 different things. On one hand, we are going to be developing biological tools. These are first mouse models. These are patient xenograft models in which we can test the drugs, but also 2D and 3D biological niches in which we can also ex vivo test the drugs. And on the other hand, we are going to be developing bioinformatics software. So these bioinformatics tools will be able to analyze the data that is generated in the project.